it off to him.
Great, and then you'll pass him the ball.
Perfect, can do. Thank you, Sherry. So today, we're lucky to have Ben Busby speak about DNA variation. Ben Busby is a principal scientist at DNA Nexus. DNA Nexus is the contractor for precisionFDA, so they're the ones who do all the work behind the scenes to make it pretty and functional. And Ben has over a decade of experience in teaching bioinformatics, and he's a well-known leader in the field. He has collaborations spanning academia, industry, worldwide. So he's really, we're really lucky to have him talk today. And prior to joining DNA Nexus, he was the genomics outreach coordinator at NCBI, and he organized over 40 hackathons. So a lot of work there, and a lot of teaching people about bioinformatics. And I think you'll agree that his enthusiasm for bioinformatics is infectious. And so again, we're really happy to have him speak today. So I will hand you the ball, and it's off to you, Ben.
Awesome. Thanks, Alexis. I appreciate it. All right. So, sharing my screen. If anybody is having trouble with anything, please put that in the chat. I am going to make a sort of spiral motion with my cursor, so if people are having trouble hearing, they will realize there is a problem and hopefully reach out for help. Anyway, so what I'm going to do today is talk, is give sort of an overview of variant calling. This hopefully will be task agnostic, because really I think what we want to think about is variant calling is really just a chain of tools, and we want to choose those tools appropriately for the larger task at hand, the larger question we're asking. And I will present this really in the context of precisionFDA, because effectively this is a system which should cost you really nothing to use, and you really have sort of free and unfettered access to do whatever you want on the system. So, and if you have sort of group members who are more technically savvy, please let them know that next week we'll be talking about building apps. I'll allude to that a lot in this presentation. We'll be talking about building apps and so on. Finally, on a logistical note, I doubt this training will last more than 90 minutes, and so we will be hopefully giving you your last half hour back. So, and I think that's a reasonably safe thing to plan on. So you can most likely plan to be out of here at 1:30 Eastern time. So that should be great. And again, any questions, please put them in the chat. Alexis and Sam are monitoring the chat. They can stop, ask me questions, and I think everybody here also has the power to unmute, so you should be able to unmute yourself and ask questions as well. So with that, I'm going to give a quick tour for those just tuning in. If you didn't catch Alexis's or Sam's awesome sessions the last 2 weeks, if you didn't, by the way, and you're interested in either RNA-seq or metagenomics, I'd encourage you to check those out. And then I'll give a quick tour of precisionFDA, really trying to foreshadow what we're doing or what we'll be doing next week. So that should be cool. Then I'll talk about variant calling with GATK, fancier implementations of GATK, really sort of our societal dependence on SAM tools. You probably know what I mean if you're a bioinformatician. I'll talk a little bit about variant annotation, why variant annotation is important. And then actually after that sort of general thing, between 3 and 4, I'm going to go on a very slow step-by-step walkthrough of an example data set, walk you through exactly how you do this on precisionFDA before I get to a final stop for question and answers. And please, again, I think it'd be great to have this as more of a discussion. So please feel free to ask questions as we go along. For example, something that's probably relevant to every single person on this call is GRCH 38 versus 37. Why would you want to use 38? Why do people use 37? that kind of thing. I'm going to talk about it quite a bit, but if you have specific observations, specific questions, specific things you want to discuss about that, please stop me and we'll just dive right into that. And if you for some reason can't unmute your mic, just put something in the chat and Alexis or Sam can go ahead and stop me. And I'm just going to flip back to the chat real quick and check. Nope. And check for any comments. I don't see any, so we will go ahead and go from here. So please feel free to reach out to me anytime. No big deal. It's bbusby@dnanexus.com. I'm also on LinkedIn. I'm pretty easy to get there. Twitter, and also I'm on GitHub at DC Genomics. Twitter in bioinformatics is kind of a huge thing. Lots of people tweet. I remember I was down at the CDC a few years ago working for NCBI, and I was giving an RNA workshop down there, and some folks said, "Hey Ben, where do we get the most cutting edge stuff?" And I said, "Well, you can check Omic Tools, which is unfortunately now defunct." And they said, "No, but where do we get the really, really cutting edge, up to the minute stuff?" And I said, "Well, ISMB." And they said, "Well, that's great, but it's only once a year. Where do we get, real-time updates on tools?" And I said, Twitter. Bioinformatics Twitter is huge. I would suggest that everybody get involved. It's really a fantastic community. People are usually really, really nice and really personal. So that's something I would highly recommend if no matter where you are in your bioinformatics journey. And your ethics officer at FDA should be able to help you with sort of the ins and outs of what you can and can't say, and your line between yourself as a federal entity and a personal entity. And all those things are navigable. There are plenty of federal employees that tweet actively in bioinformatics Twitter. So certainly a doable thing. All right, so what is precisionFDA? Basically, it's a place where you can store files and compute on them and then share your results with other people. And that's really awesome. And really, you can take advantage of cloud computing without having to do really any special backflips. And if you're at FDA, we should be able to grant you access, honestly, in a matter of minutes to hours. We can get you probably spun up on there. If you have specific questions about that, please go ahead and talk to Sam. And if you have a precisionFDA account right now, please go ahead and log into it, because I'm hoping that you'll be able to poke around with apps and tools and also maybe look at some files as we go through things. So really go ahead and think about logging in. So, this is what I actually just mentioned. You can do all of this stuff. Also, there are challenges on precisionFDA, and I'm working with some fantastic people who are actually on this call to get a precision immunology challenge going. I'm hoping that'll come out in mid to late September, maybe early October. But really, there's lots of data sets, particularly now around COVID. And, we all want to know what sort of T cell receptors and B cell receptor complements really sort of predispose people to severe COVID disease and less severe COVID disease. And really having apps for that is pretty awesome. So we'll probably have an appathon around precision immunology in September. So that should be great. All right. So somebody might want to mute their microphone. Does somebody have something to say before I jump in to sort of a quick introduction to precisionFDA? All right. Cool. So, and great. It's great that people can unmute easily. So if you have any questions, please just interrupt me. So yeah, again, I mentioned you can upload files, and also you can build apps and workflows. And those workflows are in a sort of nice GUI environment, and then you can share stuff in spaces. By the way, at the end of this, I'm going to walk you through a systematic step-by-step analysis with GATK. And anybody who wants me to share the space with them, share those data sets and those apps, it's really not a problem. I'll just need, Sam or I will just need your precisionFDA username. And if you don't have a precisionFDA username yet, you can go ahead and sign up now. It's pretty simple. You just go to precisionFDA and request an account. So I've mentioned this Dropbox thing a couple of times. We've mentioned it in the last 2 sessions, so I'm not going to belabor the point. But basically, it is a secure place where you can do analysis. And all of this is already set up for you. And thanks to Aline, as well as many other people who've been working on this system. So you can also do analyses. We'll see that at the end of this. One thing I won't talk about, but we'll talking about extensively next week, is you can import existing workflows directly if they're particularly in CWL, WDL, Docker. I've never really seen this plural, but I think it's kind of cool. I'll have to run it by Geraldine and Michael Crusoe and see what they think. And also, you can easily share stuff, and that's really, really nice. And you can make stuff public so people can see it, so on and so forth. So I think these are really nice features. And again, it's really easy to use and provided for you for free. And this we're going to talk about a lot next week. I'm going to skip over it for right now, but there's something you don't want on precisionFDA, or something that you want but you don't see on precisionFDA. It's a very flexible platform. You can throw whatever apps you want on there. And if you're even just your toe is dipped in bioinformatics, you should be able to do this. And we're going to carefully walk you through building apps on precisionFDA next week. So tune in next week. And if the second part of this is pretty simplistic for you, or you want to add apps to that space, let me know. And we can all add apps to the space I created for this webinar in next week's webinar. So that should be really cool. I really like to have hands-on sessions whenever possible. And so if we can do that advanced session as a hands-on, that'll be, I think everybody will get the most out of it. So, next I'm going to just give an overview of basic fundamental concepts around variant calling and annotation. Does anybody have any questions at this point? And I'm going to purposely, you'll see me flip my screen back to the WebEx just to triple check that nobody has
no questions in the WebEx. Yep,
no questions in the WebEx.
I did put the link to requesting an account for those that are interested.
Fantastic. Thank you, Alexis. That's great. Yeah. So it really doesn't cost you anything, and you're not going to get a whole bunch of emails or anything. So even if you have another plan or access to some other HPC, I think it would be worthwhile to get a precisionFDA account because you'll always have that as a backup, and it's easy to share stuff with people, and it's already paid for. So first, what I want to do is talk about variant calling apps on precisionFDA, what is already there, if you didn't want to make your own. So what you can do is go on to precisionFDA. And by the way, once again, if you have an account, follow along with me. So what you'd want to do is click on the apps tab. And then one thing that some people miss is that you want to click on this global explore thing. That'll let you search for relevant apps, and that's really, really nice. There are a bunch of apps there. I'll show you some examples in a minute. So you can click on the Explore tab. Here you can find apps that might be relevant to you. I'll talk about FastQC in a minute. Here are some other apps that are relevant to variant calling. And so here we have a bunch of VCF comparators. Now, pro tip, if you want to compare VCFs with that are aligned to, called from GRCH38, you should use the GA4GH comparator tool. So you just do that, find that by searching for GA4GH. Thank you, Sam, for showing me that. There's also aligners here. So BWA MEM is an aligner. We'll talk about what that is in a minute if this is sort of your first foray into bioinformatics. And there's a bunch of other stuff here too. And then we can see that there's several different flavors of GATK, and so that's a really nice thing. Some of them are 37 base, some of them are more flexible, so that's really quite nice. There's also Picard, some aspects of SAM tools, basic things that you would need for bioinformatics. But again, if you're a pro bioinformatician, you can always build them yourself on this platform. So that's a fairly straightforward thing to do. Here is one of the VCF comparison apps. Again, if you're using 38, and I highly encourage you to use 38, there's a whole bunch of reasons for it I'll get into in a minute. But if you're using 37, you can use these VCF comparator tools, or if you're an FDA reviewer and you're reviewing something somebody has done on 37. Now, as far as I know, and this is through discussions with some folks in the GRC, as well as discussions with medical bioinformaticians across the U.S. and across the world, about 70% of folks are currently on GRC H38, and about 30% of folks are still on 37. And there are some holdouts, particularly in pharmaceutical companies. But I think it's really our responsibility as bioinformaticians and as scientists to encourage people really to make that transition to 38. And references are complicated. I have a couple of slides on them, but I think GRC H38 is the way to go. And you can set up these comparisons, like I said, with a test VCF versus a benchmark VCF. Same thing with the GA4GH comparator. You can use, same thing with GA4GH. You can use a truth set and versus a test VCF. With the GA4GH comparator, you're going to want to make sure that these things were aligned and called from the same reference genome. We'll talk about reference genomes, how to get them, where to get them in a minute when we talk about the GRC. So again, I know this is the third time I've said that, but please log in and start poking around while I present this background information. So I'm going to tell you a couple of stories, a couple anecdotes, introduce you to some people who are sort of big players in this space. But before I jump in, are there any questions at this point?
Hey Ben, I'll just chime in really quick. This is Sam Westrag, and I'll say for some of you, if you haven't used your precisionFDA account in a while, you may find that it is locked. If that is the case, you can just email precisionFDA support, and they will unlock your account. Basically, they're locked after 60 days of inactivity just by federal regulations, but it just takes a quick email and it should be unlocked within a couple hours, if not earlier. Our support team monitors that and does that very quickly, and then you'll have full access once again.
Right. Yeah, that is a really good note. Thank you, Sam. Sam behind the scenes does tons and tons of stuff for precisionFDA, as well as lots of other stuff for other people. So it's great. Okay, awesome. So sequencing data basics, it's a lot of data. This is an XSeq by Illumina, and it's this thing can pump out something like 44 human genomes in 48 hours. Transferring data can be an issue from these machines. It's really amazing what these things can put out now. And so just for people who are complete noobs to bioinformatics, and kudos to you for being here if you are. You're starting your journey. So this thing will generally pump out millions and millions of reads that are about 300 nucleotides, usually paired to another read that's about 300 nucleotides. So that's sort of the state of play. With short read data, about 90% of the data is coming out of one of these machines. Next generation sequencing data is coming out with one of these machines or a similar machine. Although we still use panels and chips quite a bit, particularly for large population sequencing. This is a global screening array chip. So this will sequence about 1 million variants. If you buy 1 million of these chips, you can get them for $30 a piece. So you could screen 1 million people, get 1 million variants off of them. And a lot of custom chips now are based on this chip. Again, this is not an endorsement of Illumina. They're just a huge player in this space. So their stuff is worth mentioning at this point. So if you can imagine, obviously, 1 million sites is only a small fraction of the genome. So what we can do often is impute between different sites in the genome. And so this is a little bit of a tangent, but it is possible to tell other things, particularly when we have pretty good knowledge of haplotypes. And also when we have phase data. So what is phase? Phasing is when you know which nucleotides are on which strands. So that's a useful thing. And then you have informative sites that can be, which copy, and you have an informative sites that will tell you which haplotypes you have phased. These are similar to graph informative sites, and Ben Langmead at Hopkins has been doing a lot of work on that type of stuff lately. So I mentioned short read sequencing. I'm not going to go into this too much farther. And then now we have long read sequencing. I think most of you know, probably 95% of you know, this kind of comes in two flavors. Obviously, top left, this is PacBio, now CCS with their barbell technology, reads the same thing over and over again, and then comes up with this very clear consensus. The other one is Nanopore. Now, so with CCS, you're getting super clean, long reads. If you're establishing a new genome, part of the human genome, which is very ambiguous, these are wonderful to have. Nanopore, you can go fast and cheap and big and do huge sequencing projects. It's a very exciting time, I think, for long-read genomics. Reference sequences. I've already belabored this point, but I will go into it one more time. So the Genome Reference Consortium consists of a bunch of folks, including EBI, NCBI, Sanger, and WashU. So basically, they have put out GRCH37 as well as GRCH38. Why would you move to 38? Let me give you a couple examples. So in 37, centromeric sequences are not modeled, so they will become a sink for RNA-seq reads, an aberrant sink, and your RNA-seq will be off. So a great reason to move to 38. Also, there are regions which are just not resolved in 37 that are resolved in 38. Finally, there are alt regions, many more alt regions in 38 than 37. So for that reason, and alts are sort of local graphs of regions that don't make sense to be represented by one representative genome. So, and I'll talk about graphs and why they're important a little bit later. You can think of GRCH38 as having a few mini graphs in it. So, and then now we have 1,000 genomes. As I think most people know what 1,000 genomes is. If you're new to bioinformatics, basically this is, we as a community, bioinformaticians, took 26 populations, 100 people each, and actually did a whole bunch of chip-based sequencing or chip-based calling. And then now we have 30x on GRCH38 done by New York Genome Center. And that's actually the data that I'm going to walk you through at the towards the end of this presentation, walking through calling variants from there. Although I will tell you honestly, they've already done that. If you're a bioinformatician, you already know that because you can see that you can just grab that. And I'll talk about how to grab that into precisionFDA. Also, by the end of the year, we're talking about having Jupyter notebooks and some other really nice features in precisionFDA. So you should be able to do some really cool tertiary analysis stuff, variant annotation, etc., right in the precisionFDA platform. Sam said I could say that. So that's an exciting thing. This guy. So this is Justin Zook. He's sort of a celebrity around FDA. Most of you probably know who he is. You see a super tall, lanky guy walking around who's super nice. That's Justin, and or possibly the other Justin that's in his lab. Sorry, that's a bit of a joke for those of you who know him. But anyway, so this is Justin Zook, and he and formerly Mark Sallet, they put together this collection with a whole bunch of other people called Genome in a Bottle. And basically, Justin published a paper a number of years ago now where basically he demonstrated that about 80% of the genome is really sort of very easy to map reads to. 10% of it is kind of okay, and 10% of the human genome is kind of a mess with respect to mapping short reads. And so those are areas of the genome that are really under close observation. But if you really want a truth set, a very clear truth set from about 8 human genomes, really look no further than Genome in a Bottle. And so you can just find this by Googling Genome in a Bottle. That should be a helpful thing. Also, if you're really new to bioinformatics, please title your files like this. So you don't have to use the camel case, but underscores, never spaces. That's just a public service announcement. Okay, and then haplotypes. I mentioned haplotypes before, and now we're starting to know more and more about haplotypes for a bunch of different populations. So the Haplotype Reference Consortium is a little bit biased towards Caucasians, actually very biased towards Caucasians. And then, but there's more data coming online, again, mostly Caucasian, from UK Biobank, but then from a whole litany of other biobanks across the world. And I think that's very exciting. And so my hope is that in the next year or two, we'll really start having a good idea of haplotypes from around the world. There's already these haplotype imputation servers. If you're interested in that, go check them out. I want to talk a little bit about pangenomes. I don't want to belabor this point, but basically the idea of pangenomes is instead of aligning everybody to a single reference point, we can make pangenomes of a bunch of people. Often these are expressed in graphs. This is a very simplistic graph of GRCH38 running through a bunch of primate genomes. But anyway, so if we can align to graphs, then we can sort of see where people's haplotypes fall in a number of groups. And as you probably have intuited, that's similar to looking at an individual versus a group of alts, hence sort of the allusion to mini graphs. If this is something you're deeply interested in, I'm helping out. Baylor College of Medicine, with the help from DNA Nexus, is going to be running structural variant and pangenomics hackathon October 11th through 14th, and that'll be all virtual. There's only 100 spots max, so if you're interested, please send me an email, and I'll make sure you get the sign-up materials. We'll get you in there soon. Any questions right now? I thought that I heard somebody say something. All right, yeah, so you can check out the human pangenome. Specifically in that, I'm going to be taking these 30X, hopefully with the help of Giuseppe Narcisi. I'm hoping to lead a team taking these 30X 1,000 genome sequences that were generated by the New York Genome Center and making 26 reference graphs. So the idea is to have the first reference human graph genomes for 26 populations from around the world. And I think that I'm hoping that sort of kickstarts us thinking about really aligning sequences to appropriate references and diverse references. That's my personal hope. So short variants. So there obviously variants kind of come in two flavors, short and long. They are split up by a relatively arbitrary cutoff of 50 nucleotides. So we call anything under 50 nucleotides a short variant. We used to call them SNPs. Anything above that is a long or structural variant. Indels are complicated. Short indels are complicated. They're somewhere in the middle. But anyway, so DB SNP houses short variants. I think it's sort of an authoritative source for. It's the most authoritative source for short germline variants. Cosmic is a great catalog of somatic variants. And DB SNP is free for everybody. Cosmic is free for non-commercial use. So that's two fantastic resources. So, and the reason I show this is because this is something I often think about. If I'm going to say a variant is a germline variant, that's a real variant, I want to be able to see it in DB SNP unless I'm looking at some sort of ultra rare, really funky phenotype, right? Like, you should see this. And what we're seeing more and more are combinations of relatively common SNPs that lead to chronic disease. But really, sort of as a sanity check, they should be seen in DB SNP. It's a database that's growing very rapidly, so something to check out. Structural variants, I mentioned that we're doing this thing with Baylor, something that I think is very interesting and getting very related to human disease. And one thing that's really crucial with structural variants, and if you remember anything about structural variants from the talk today, is remember that it's very hard to overlay long structural variants. And so a Note SV is taking kind of a crack at that in terms of overlaying big structural variants that might have kind of fuzzy ends. And that, I think, is going to make it possible for places like DBVAR really to reach into the future. So that's a cool thing. And thanks to folks at Elixir really for promoting that. Parliament 2 is on precisionFDA. It is a great comparator tool for structural variant calls. So this is really for the bioinformaticians in the room. But if you need to do that, Parliament 2 is already there for you. And so you can come up with consensus structural variant calls from a variety of structural variant callers. So this I think is a really nice tool. If you were to ask, I'm sure we could put CNV kit on there pretty easily. In Ben's personal opinion, hopefully the term CNV will be eradicated in the next 5 years, because really what CNV means is we don't know exactly what kind of structural variant it is. And I would like to think that we'll have better algorithms to characterize structural variants in the next 5 years. And then also there are algorithms for functional prediction. As Jupyter notebooks start to come online, we'll have more and more. But here, you have basic SNPF. If you need sort of basic, super basic functional prediction, whether SNPs are coding, non-coding, what they do, sort of quality, frequency, that kind of thing. SNPF is pretty it's pretty standard, pretty easy to use, and pretty flexible. So that's a really simple variant annotation program for those of you following along. And that's a sort of an example of basic functional annotation. I mean, VEST is really good. There's a couple of other really good functional annotators. Still, typically, they're not as accurate for health and disease prediction as clinical annotators. And with the KG people, that's critical assessment of genome interpretation, basically assessing people predicting what variants do in the context of disease, would say is you should be using clinical annotators in concert with functional annotators. And so that's something I think to think about. So I think that is what I wanted to say generally in terms of background. So then let me go through what is a basic generic variant calling workflow. This is something we sort of do all the time. So I'll go through sort of what is a platform-independent variant calling workflow. Talk about this a little bit, and then we'll pause for questions. We'll take a little coffee break, and we'll come back and work through this kind of. We'll work through the first two steps of this on the precisionFDA platform. So first, you do pre-processing. You're going to want to clean up raw data, remove contamination, and removing and trimming. So tools for that, the most common tool is going to be FastQC. You're going to take a look at what your stuff looks like. Usually it's pretty good, but sometimes there's some systematic errors. It used to be that you'd see big strings of G's at the end of reads, that kind of thing. It's improved lately. But you want to check things out, make sure you're generally okay. Quality scores, my feeling is that you don't need to be that specific. RNA-seq, you probably don't need quality scores at all. That's Ben Busby's personal opinion, not the opinion of DNA Nexus. But then for mapping and calling, for mapping DNA-seq, obviously RNA-seq is much more complicated. Alexis went through that 2 weeks ago. You can catch the recording. But for DNA-seq, most people use BWA-MEM. It works really well. And also what's really coming online, if you're looking at large cohorts, is Google's Deep Variant. If you're interested in that, please ask in the Q&A period or just send me an email. I'll point you to the links, or maybe Sam or Alexis will put one of the links in the chat. That's all coming online. By the way, there's some other really cool stuff. And then Deep Variant's a caller anyway. And then long read mapping is a bit more complicated, and I think it's beyond the scope of this discussion, but if you have questions about that, Deep Variant is a caller, as is GATK. So these are the most common. GATK also has derivatives. So there's a derivative called Sention, and actually there's a flavor of Sention already in precisionFDA, but Sention is basically a deterministic GATK3. And then there is also a NVIDIA derivative called Parabricks. That's a cool thing. If you want to try that, let me know. I have some trial and educational licenses. We can talk about it. So the folks at NVIDIA, in my personal opinion, are really nice, and they let people use stuff. Sam, correct me if I'm wrong, but I'm pretty sure we don't have GPUs on precisionFDA. But if you're interested in GPUs or doing machine learning or that kind of stuff, we can talk. Maybe I can help you find some way to do that. And then basically, eventually, that's a little bit out of scope for this talk. You're going to want to annotate your variants, extracting known things. And again, think about using both clinical. There's probably now, ClinVar is the most commonly used one, but there's a bunch of them for cancer. So think about using a clinical annotator or multiple clinical annotators, as well as several germline annotators. Several functional annotators. So thinking about like VEST or REVEL, thinking about like what the function of the amino acid is in the protein and trying to predict that. In concert, KG has found that that is really the most effective way to predict functional variant consequences in the context of disease. So making sense of the results, obviously custom scripting, but here's something I would tell you. I have a friend named Louis Gear, and Louis, when I was a young bioinformatician, Louis helped me think through some problems. And, here's the thing he would have said to me. He said, Ben, when you run a gel, I was a biochemist like 15 years ago or something. He said, when you run a gel, what serum do you use to block? And I said, well, I like fish serum. And he said, why do you use fish serum? I said, well, because it works well. And he said, well, what are the molecular components that make it work well? I I said, I don't know. And I think the same is kind of true with thinking about bioinformatics tools is, really, they're intermediate reagents. What you should think about is whether the analysis is clean, whether variants are coming out in agreement, whether you're finding haplotypes that make sense. that kind of thing is what you're really looking for at the end of the day here. And so really just asking, for those of you who are primarily biologists, asking bioinformatics questions is the same thing. It's just with a computer in front of you. So that's what I wanted to say about that. And then in the next session, I will get off my soapbox and actually go through a systematic methodological review of actual variant calling. But I thought it was important to give you sort of the societal bioinformatic context of what we're thinking about and say some words about thinking about problems. By the way, points, I'll give anybody a Snickers bar next time I'm at the FDA, if anybody knows what this is.
No idea.
Anybody? All right.
There's a guess that it's a coffee bean roaster.
Yes. Somebody
Heather
knew that in
Harbottle's
the
the
chat?
winner.
What's that?
Heather Harbottle is the winner of today's question.
Heather, I think we have a meeting on Friday, but I'll try to figure out a way to get like some sort of candy bar your way. That is an SR500 coffee roaster. So if you get to some sort of point with your coffee consumption, you can start roasting on your own. This is about $150, and it will roast coffee. It will roast 3 pots of coffee, like 3 full-size pots of coffee in about 5 minutes. So I just wanted to share that with you. Okay.
There's one question in the chat box.
Fantastic.
They're asking, is there a feature in precisionFDA to integrate external database for variant calling and to predict drug resistance? And their specific case is HIV.
Wow. So I know that there are drug resistance. All right. I'd have to dig into the question a little bit more on, to give a sort of slapdash answer. What I will say is that there's Farm GKB there's an implementation of that in precisionFDA right now. So you could look at that. I think that in general, drug resistance is a tricky question, but more and more cool data is coming online. So there was a data set that came out of the University of Tokyo that is in the NCBI SRA that came out, and DDBJ obviously, that came out in April, and it looks at a whole bunch of cancer cell lines. I think, if I remember off the top of my head, lung cancer cell lines. And then they've tested each of those with 100 drugs. So really, there's a couple of databases for it, but, databases tend to be, for that kind of stuff, tend to be pretty general, if I understand your question. Large data sets like that tend to be a little bit more specific. And really, I think at the FDA, I think that you're going to have folks submitting stuff, with that stuff. And I think submitting stuff with that stuff, submitting applications, with drug efficacies. And I think it's worth doing some thinking if you can find any evidence that would say that somebody is resistant to a particular drug. Did I get anywhere close to answering that question?
So I think a simple answer, and he says yes, I think at least from my use of precisionFDA, if there is a database on the internet that has the information you want, you can pull it into precisionFDA. And so apps on precisionFDA have access to the internet. And so you can modify, let's say, an existing app that's pulling in a different database and pull in the database that you want. And so you can also, if it's a private, if it's like proprietary data that a sponsor sent, you can pull that in and and use that without it being a public app as well.
So that is absolutely true. Yeah, that makes a lot of sense. And I would add to Alexis's comment just to say that this is going to be even easier once Jupyter Notebooks come online. I mean, it is really, there's some really nice platforms that are pip installable to do that kind of thing, as well as, like Alexis said, just being able to bring in databases from the internet. So it's going to be, I think, a very exciting time, not only for bioinformatics in general, but specifically for precisionFDA the next few months. Are there any more audience questions? I haven't looked in the chat specifically. For some reason.
I think that's the only question.
And again, for the bioinformaticians in the room, I'm going to try to intersperse sort of anecdotes and tips and tricks for more advanced bioinformatics with sort of basically walking through the pipeline. So hopefully we'll hit everybody and make things interesting for whichever, wherever you are in your bioinformatics journey. All right, great. So I'm going to go ahead and share my screen, but I'm not going to go back to the slides yet. I'm going to go to precisionFDA, and I'm going to go, and so this is the space. So when you, I can't click on spaces because you'll see other spaces that I have access to, and there are some non-FDA people here. Again, kudos to my old NCBI folks for coming to this. That's awesome. Anyway, so there's some non-FDA folks, so I can't show you the general spaces and what it looks like. But if you're interested in sort of poking around with the apps and the stuff that we go through today, I am totally happy to give you access to all the files and the apps that you would need to recapitulate what we did here. So as long as you have a precisionFDA account, you can send that to me, and I'll get you on here, and you can go through. And really, this isn't it's stuff that I've uploaded pretty recently, so it's not a big deal. Don't worry about hurting anything, any of that. So you can really kind of kick the tires and mess with some things. So with that said, I will go back to the presentation. And let's talk about a step-by-step variant calling workflow. I'm going to try to make this simple. It should take about 20 minutes, give or take, to go through this. Again, intermediate reagents, pretty straightforward. So the first stage really is to import data from an outside source. In this example, we're going to use the 1,000 genomes from the New York Genome Center. And we're going to use the precisionFDA URL fetcher. I find that to be a good thing. Now, in general, outside source data is going to come in a number of formats. So sort of if you get the data from somebody else, the most common format is going to be FASTQ or BAM. For those of you who are very new to this, basically FASTQ is minimally processed next-generation sequencing data, basically sort of the lingua franca of NGS data. So you get that. BAM is just that mapped to a reference genome. Now, the only trick with getting a BAM from anywhere is what reference genome was it mapped to. And good places will show, I'll show you the documentation with New York Genome Center later, will tell you, they will actually give you a link to what reference genome you map to. And when you do mapping, if you're going to share your data with anybody else, you should provide a link or some access to the reference genome data you use. There's a ton of reasons for this. Basically, downstream tools won't work. And also, if you don't have the same exact reference genome, there are big errors, little errors, all kinds of errors. But basically, also in bioinformatics, accessions within these genome references come in 3 flavors. They can be summarized EBI, UCSC, and NCBI. There's pros and cons to all of them. So it's really important to let people know which one you used. So anyway, and then the other data formats that you may get are CRAM format, which is popular with EBI and the Broad, and then also SRA, which is really sort of a columnar database. And many people use something called FastQ dump, but if you're doing relatively informal work, you can also do pile-ups directly. That's beyond the scope of this, but in a former life, I made lots and lots of webinars on that, so you can look them up on the internet. And I'm happy to send you links to that. Those are basically the formats things will come in. The format for what we're going to get today is CRAM format. And CRAM is easily convertible to FastQ or BAM as long as you have the exact reference genome. Otherwise, things will not work. So make sure you get the exact reference genome. And if the sequences have been aligned, it's great to go to BAM. BAM is, you can do lots of stuff from BAM, including calling variants. We're going to use GATK. It's pretty straightforward. And then what I would do after is do something like take this VCF, you can put it into IGV, or you could look at a gene locus just with grep or less or whatever, and that I find really sort of useful. So really, I'm very excited. Honestly, I'm going to tell you guys, honestly, when I first heard about this data set, maybe 18 months, 2 years ago, I was not super excited about it. A guy named Steve Sherry said, Ben, NHGRI just funded the New York Genome Center to sequence 2,600 whole genomes. And I said, well, that's great, but what we really need is a huge biobank. And of course, now we have the All of Us project coming online, and of course, UK Biobank. But I think actually this is really cool because we have really sort of a diverse reference set that almost every bioinformatician ever has worked on. And so, all bioinformaticians that do human work. Obviously, there's some amazing plant and bacterial people who don't care about this data set. But anyway, and the really nice thing is there's VCF for this already. So you can grab the VCF and then go ahead and proceed with those tertiary analysis. Also, really sort of do tertiary analysis. That's a nice thing. And here for individuals, you can. It's really easy just to get URLs. Now, a really nice thing about having simple to resolve URLs is in precisionFDA is you can just use the URL fetcher. And so this is a fairly straightforward thing to do. Although I'm going to put a couple of tips and tricks in here. So I like to use the high memory things when I upload things because some of these files are really big. So I usually use not high memory. I apologize. High disk. Usually I use high disk 8 for the instance type, which I think is pretty useful. And then you just pop the URL in there. That's great. And then rename into makes it really easy. So this particular thing is NA12778. That is a person, I believe, in Utah. And so you're going to put you could put NA12878 or whatever else you want here. And then it'll rename this to that. So yeah, and then it's as simple as clicking Run App, and it will go ahead and upload for you. It's really quite nice. You go and do something else for a few minutes, come back, and your data is there, and you don't have to worry about a whole lot about that. Also, Alexis Norris, who is on the call, has built a URL fetcher that will work with directories now, which is awesome. So if you needed, I know this is an example that isn't particularly pertinent, but just for fun, if you needed all bird genomes, you could totally do something like that. Or if there were a set of, say, folks or cell lines who were particularly resistant to a particular drug, you could download those profiles as well as a directory. So that's a really nice thing you can do with the URL fetcher. And this is I think the URL fetcher is really the easiest way. You can also upload files from local, but I think nowadays, these data files are getting bigger and bigger. They're more and more numerous. It's great to go from cloud to cloud and not touch your local computer. Then, you don't risk gumming stuff up. It's not so much of a mess, so on and so forth. So that's one thing I prefer to do is really only transfer, if I transfer at all, sort of like processed files to local. And honestly, I try not to put things on my local machine at all whenever I can because it's really not good security practice to do so. Any questions about getting data with URL fetcher? Once, and twice. Okay. And then resulting files are in an area where the app ran. So I showed you files in spaces before. So this is file in space if it hasn't timed out. Yep. So these are shared area files. They're in this area. And so then these will show up in this space. So if I give, if 24 of you ask me for access to this, I'll put you in the space. And then if you run any of these apps in the space, you can just click on them, run them, whatever, blah, blah, blah. If you run any of these apps in the space, then your derivative files will end up in the space. It's also very easy to do things like add a folder. So I can say, reference genomes, and I can create a folder, and then I can simply pop things into that folder, and that's really quite nice. So that's a nice feature, and right. So going back, so that's files. So you could convert to FASTQ. I put this in mostly because you probably wouldn't convert an aligned thing to FASTQ, but some people say, well, what if I need to go back to FASTQ? Then you can, if you want to. Often data is only in FASTQ, or sometimes it's mapped to a reference genome that you don't like, right? So I've extolled the virtues of GRCH38, but maybe there's some old data that's super relevant to you that's on 36, and so you want to remap it. There's some functions, liftover, remap, which are UCSC and NCBI respectively. If you don't want to mess with that, you can just dump the FASTQ and remap it with BWA mem. Pretty straightforward thing to do, not a huge deal. And you can always go cram to FASTQ. Basically, any modern BAM should go back to FASTQ pretty easily. just again, make sure you have the appropriate reference file. So that's always the big deal. And so you really want to, when you put out any kind of alignment, tell people about the reference file. It's really helpful, and otherwise people can't really do too much with it. So here's alignment with BWA MEM. Instead of sort of laboring over this point, I put up the specs and workflow of New York Genome Center. I think they did a good job of outlining this, GSFA, and some other awesome people that are over there, worked on this project. Kudos to them. A lot of fantastic professionals. So this is BWA MEM, which most people use. So there's also HiSat 2, which is also on the platform. It was demoed 2 weeks ago. Yeah, Alexis used HiSat 2. So it was demoed 2 weeks ago as an RNA-seq tool. It's primarily an RNA-seq tool. You can use it for DNA if you want to. So if you had like a project where you have some sort of targeted sequencing and you want to do some stuff quick and you need to be super flexible, HiSat 2 is a reasonable idea there. There's also a tool I used to work on called Magic Blast. It's for really informal analysis. It's great. It's basically BLAST for genomes. And so that's really quite nice too. Actually, sorry, BLAST for next generation sequencing. We've always had BLAST for genomes. Not always, but you know what I mean. Okay, great. So there's that. And then, but what I think really is the meat here is converting to BAM. Also, you know, speaking of New York Genome Center, the person who at least used to run it is Toby Bloom. And Toby Bloom is a smart woman. And I was on a panel with her, and I remember, I will never forget this statement. She said, when are we going to start trusting people to make intermediate files, right? So the thing is, somebody hands you a BAM, and you have reason to trust them, you know, it's probably fine, and you probably go from there. There's really no reason everybody has to go back to FASTQ for every single experiment. So just something to think about. Now, not everybody aligns to the same reference unit, so that's an issue. But when you, and you'll see this in the spec from New York Genome Center, basically they're like, everybody who works on this project has to use the same reference unit. That's probably something you want to think about in your projects as well. And honestly, like if you're working with a number of companies or whatever, please try to convince them, go to 38 and use this with alts. And that's typically important. Otherwise, we're doing sloppy bioinformatics. And I mean, I don't want, as an American taxpayer, I don't want drugs to cost $800 billion. And so I think by avoiding, in my personal opinion, by avoiding sloppy bioinformatics, we can probably help that problem just a little bit. And that's really in the realm of our expertise. Now, that's obviously a personal statement. But anyway, and many of you may have strong feelings about that, and I apologize if I've offended anyone. Anyway, so that's why you should go to BAM. And then after that, run GATK. And the nice thing is the GATK4 Best Practices Toolkit is everywhere. It's on DocStore. I mean, the Bro distributes it. You can, if you have, I'm not really supposed to talk about DNA Nexus, but if you have access to DNA Nexus, you can run it there. You can run it on precisionFDA. You can run it anywhere you want. That's really great. And the haplotype approximation is all right. There's some other great haplotype predictors. If you're really interested in haplotypes, take a look at them. And then also, I would say that it's important if you're involved in research at all, don't be scared of structural variants. Find a simple to use structural variant tool. I think Parliament 2 is great, and call SVs as well. So I think that's a nice thing. GATK, the reason I'm not live demoing it is fairly simple to use. Really, it shouldn't be a huge deal. Now, if you're a super sort of dyed-in-the-wool bioinformatician, you got a lot of complicated analyses to do. I mean, that's something that, you know, I mean, you can talk about GATK and what it does and so on until you're blue in the face. But I think right now it is sort of the that and derivatives of it are sort of the standard for small to medium amounts of genomes, unless you're talking about huge projects. And if you're talking about huge projects, again, deep variant is coming onto the scene, and it's really designed to think about cohorts of 100,000 people. And I hope, I mean, we do some joint calling, and I think joint calling, this is supposed to be fairly basic, so joint calling is beyond the scope of this discussion. But, I hope we can really get to a place as a community where we're sort of doing joint calling for populations of interest on the fly. That would be really, really nice. So that's really what I wanted to say about GHK and deep variant. And then, whenever I do one of these analyses, I like to do a sanity check. I'm kind of interested in a gene called anchorin-1. It's got some funky short isoforms. That's kind of neat. It's neat to think about, like what intronic SNPs can potentiate those isoforms, so on and so forth. So I would advise everybody, look at your favorite gene. When you get a new set of VCFs, look at your favorite gene. You're going to know a lot about it, right? Like, I mean, at least if you've been a biologist for a while, like you'll know, like where different domains are and so on. And so you'll probably have an idea of where different coding SNPs lie, maybe, what intronic SNPs might be important, that kind of thing, promoter regions, etc. So yeah, take a look at that. I think that's a neat thing to do, and that can always be done with grep, or if you please don't fit a whole please don't try to put a whole VCF into Excel, although nowadays you can, but be careful with that. We know that it renames gene names to date sometimes. But anyway, but if you can control F, and look at your favorite gene, I think that really brings us down into reality, and you're going to know things about it which really bring biological knowledge to bear on your bioinformatics, which is something we all want to do. I mean, if bioinformatics was simple enough for robots to do it, we would have figured that out already. And it's not so much because the biological space is not sort of rigid. It's fairly fluid, pun sort of intended. And so really it's important to think about whether things make sense in the context of your phenotype or disease or drug susceptibility, as somebody was wise to point out. So I can share this space with you if you want. Please poke around. Please explore. That should be pretty exciting. This is something I already showed you. Yeah, I'd like to thank Daniel Quang and Sam Westreich. That's a picture of him there. And oops. And these are Alexis's dogs. This is a picture I got from the internet. So yeah. And the reason I showed this picture of Sam is this is crazy. Like I saw this myself with my own eyes. So he took, this is a Manhattan plot that you'd get from a GWAS, and he actually made this viewable with VR goggles. So check out this blog post. Just Google like Big Top VR Genomics. You should be able to find it. Google this blog post, and you can actually see how you would use VR goggles to view Manhattan plot in this Big Top virtual reality platform. So anyway, that's a cool thing he did. Oh, and then there's a blog post here by Daniel Quang about topics in bioinformatics and AI. And Quang is a young fellow who does some awesome stuff with machine learning, and he's fun to bounce stuff off of and think about. And he teaches me about NVIDIA drivers, specifically CUDA drivers, that's complicated. Yeah, final question and answer session. If you have a complex question, please email me, email Alexis, email Sam. And there's one more session, which is building apps and workflows. And then we'll talk a little bit about scripting and Python, R, and Jupyter's coming. And we're very excited about that. And I want to talk to the bioinformaticians in the room. I can't wait. I think Alexis and Sam and I will all be co-presenting the last session, and I can't wait to talk to you guys about Jupyter Notebooks. We'll probably put up a Jupyter Lab session of some stripe so we can have something to show you guys, and I think that'll be really neat. So with that, this is the final Q&A session. I promise to let you guys go at 2:30 Eastern Time. We got 7 minutes for questions. Let's see if we can cram all the questions in here. If you're eager to get out of the room and this is your 7th Zoom meeting of the day and you just can't take it anymore, shameless movie reference, then have a great one. If I never see you again, have a fantastic life. Most people at FDA I'll probably see again at some point, so I can't wait to work with you further. And now we will go to questions.
And if you want to unmute to ask a question, feel free to do that.
Yeah, go for it.
I'll just mention quick that Sam is also a DNA Nexus, and he's our point person for basically anything, and so he's the magic problem solver of everything. And if you ask a question, he'll probably answer it. I am not with precisionFDA. I'm a reviewer in CVM and a bioinformatician, and so we use precisionFDA exclusively for receiving sponsor data related to sequencing data sets. You know, a recent submission had over 0.5 terabyte of data, and so for us, that was a real game changer, and for the sponsor to be able to do that electronically, especially now in the COVID environment where we don't want to be going into the office to receive a hard drive. And we also use it for running apps, so running some analysis. And the reason that we have access to other clusters as well within FDA. But one nice thing about precisionFDA is that when we build an app on there and we run an analysis, we can share that app with the sponsor, and they can use that same app. And so it just increases transparency. My group has a lot of smaller kind of biotech startups that we work with, and a lot of them don't have a bioinformatician, and so it really reduces the barrier for them to be able to do bioinformatics. And so it's been a real success story for us at CVM.
Yeah, I have a question to Ben. So you talked about, let's see, the NYGC 1000 genomes. Is this something that, is this something that you can look at related to looking at some kind of visuals for some genes, or it's not? What are there any efforts, do you think, that DNA Nexus is thinking about doing something for doing some visuals on precisionFDA?
So, DNA Nexus has some integrated visualizations on its platform, but I will let Sam Westrike speak. I think he's still on here. I will let him speak to what we are planning to do, because that is his thing.
Yeah, so right now we don't have a lot of visualization options on precisionFDA directly. You can always take the files off and you can load them into something like IGB if you want to view them. We do plan on integrating that at a date to be determined in the future. I can't speak exactly to when it is because it's not on the roadmap for the next couple months, but it is something that we've tossed around in discussions and we're going to look at. And that would probably be bringing in like an instance of IGB that you can just load files into and it would run in browser and show up right in precisionFDA. That's the vision for it. And so, the great thing about precisionFDA is because everything is file and app based, you can always export files at any stage off of the precisionFDA platform, you can load them into any other visualization tool that you want. And then if that's an intermediate file, or you say, I want to pare down these results further, you can always just throw the file back up onto precisionFDA to do more steps to it.
Now, one thing that is in the pipeline, as I mentioned earlier, is Jupyter Notebooks, and those I've heard, although I can't guarantee anything, of course, I've heard that by the end of this year, Jupyter Notebooks will be up there. And when I want to visualize genomic secondary or tertiary analysis, I typically do that using notebooks these days. and they are interactive. So I think that's going to be a really attractive thing. And then if you tune in next week, we'll almost certainly talk about data frames and interacting with data frames, and then that makes visualization in Jupyter Notebooks even easier. Alexis, you have a lot of experience with that kind of thing too. Do you want to talk about that at all?
Yeah, so I'll just add that, so I use R because R is, so Jupyter's primarily what Python users use, although it works with R as well. I come from preferring R for data visualization, and so I use RStudio Shiny apps. And the beautiful thing about a lot of the data visualization, whether you're using R or Python, is that you don't need cloud computing for a lot of it. And so typically what I do is I do all my processing on precisionFDA or an HPC cluster, and then I download the small final results, whether that's, it's usually a table, and then I parse that and visualize it in R. And if you want to go back to the session 1, so in session 1, I go through our use of our Markdown notebooks to do final analysis, and then also our Shiny apps to do data visualization for RNA-seq data. But that runs on your local computer.
Yeah, I see. So you go from precisionFDA to your local computer to use R Shiny to do some visualization.
Yeah, and I really, I'm used to doing the final data visualization on my laptop. And I've just got your standard Dell FDA issued laptop, and it works fine.
You could certainly build, if you had a standard visualization that you like to see a lot and the R script that generates is pretty consistent, you could always run that R script as a precisionFDA app as well. You'd provide the R script as an asset to precisionFDA, and as long as the inputs and outputs are specified when you run it using the R script command, you could do it that way and generate images if that's something that you're after as part of like a final output from a pipeline. Just keep in mind that currently with the way apps are set up, it wouldn't be interactive code. So it's not that you can be like, oh, I want the barrier to be, 5 pixels higher. But you certainly can run R scripts in an app on precisionFDA.
Yeah, and I suppose if you can have this app, then you can have like a general one for visualization. Let's say look at the genes on a chromosome, so you could probably use this app on precisionFDA to run it, and then takes you to that visual and do whatever.
Yeah. If it just runs and outputs a visualization, either an image file or HTML or something like that, then you can just open and view it once it's finished running. Yep.
Actually, and with most of the apps I already showed you, you can have target regions. So if you're really only interested in a particular target region, or if your app, is looking for enhancers and you're only interested in enhancer regions or something like that, you can usually restrict to that in the app, and then you have a relatively small file and a small area to look at. And the nice thing is the data will be there. So if you ever have an impetus to look at it again, if you're like, well, you know what, that was cool looking at the answer regions, but I want to look at this, that, or the other thing, then there's just no reason you can't go back and with sort of one click look at the entire file.
Yeah, that's great.
Cool. All right. Are there any other questions? And kudos to everybody for hanging on so long. Trivia question. Does anybody know, so we talked about Python and R in Jupyter Notebooks. Does anybody know where the JU in Jupyter comes from?
No idea.
Really? Seriously? Alexis, you don't know?
I don't do Python.
I
avoid Python like the plague.
It has nothing to do with Python. Yep, it's Julia. There you go. Thank you, Joseph. Yeah, it's Julia. And I was talking to somebody last night, and they're like, "I code in Julia," and I was like, "Wow, that's awesome." So yeah, I mean, coding languages don't die. It's just that there's like 10 people that use them, and everybody's like desperate to know how that works. So you've seen examples earlier this year. And I stole that quote, by the way, from Hadley Wickham, who is he's sort of an R celebrity these days. Anyway.
Oh, and so just one more thing. If you're interested in R in our studio, I put together some really informal small group trainings, and there's, I think, 3 of them we're doing in September. So they're not going to be large like this. It's going to be smaller, more hands-on, as much as you can do that in the virtual world. So if you're interested in those, please email me, and I will add you. So it'll be just kind of informal. Here's basic how to use R in our studio. This is a Shiny app. This is an R Markdown notebook. If you're interested, let me know. And we'll be doing Tidyverse, so that's Hadley's approach to R.
Yeah, if you've ever seen like percent signs with like less than signs in the middle of them and you had no idea what that was, that's all Tidyverse stuff. So if you're an R user and you're confused by that, it is really very useful in my opinion.
It's great for learners. I think it makes the code much more readable.
Yeah, and it's really easy to manipulate data frames too. It's a little bit more native, I think. All right, awesome. Well, I hope everybody had a great time and that you're happier than when you came into this meeting. If you're not, let me know. We'll see what we can do to fix that. And I hope you all have a great rest of the day and great rest of the week. And I hope to be talking to you guys next week, particularly the bioinformaticians on here about how to build apps. And if you have any questions in the meantime, you know where to find all of us. All right.
Thanks, Ben.
Thanks, everybody. All right.